Right. So, chapter 3, I have titled The Basics of DNA and Cell Division. So, we're going to go over some DNA replication, some protein synthesis, mitosis, meiosis, all that good stuff. All right. So, there are two types of cells. These are different from classes. The two types of cells are prokaryotic organisms and eukaryotic organisms. So prokaryotic organisms are single-celled organisms. They do not have a nuclear membrane or distinct organelles. You can think of organelles as kind of like the components or the compartments of the cell or the organs of the cell. The prokaryotic organisms have single-stranded DNA that's contained completely within the cytoplasm of the cell. The cytoplasm is kind of the liquid that helps the cell keep its shape. Prokaryotic organisms are the very first life on Earth. And we see evidence of prokaryotic life going back about 3.5 billion years ago. So, remembering back from chapter 2, the planet itself is about 4.6 billion years old. And we see the first evidence of prokaryotic organisms about 3.5 billion years ago. So, of course, this is a class on humans, and humans are animals. So, the vast majority of this class, we will be talking about eukaryotic organisms. So, eukaryotes are multi-celled organisms. They do, in fact, have a membrane-bound nucleus, and that nucleus will contain all of our genetic components, all of our DNA, all of our chromosomes. So for humans, we have 46 individual chromosomes or 23 pairs in every somatic cell. We'll talk about what a somatic cell here is in just a moment. But just know within every single skin cell, every organ cell, every tissue cell, every bone cell, within the nucleus of those cells, we have 46 individual chromosomes or 23 pairs. So I don't know why my slides keep going forward before I'm ready today. All right, so the DNA that's within a eukaryotic organism is a double-stranded helix-like structure. And we have two different types of DNA. So we're going to have nuclear DNA that's contained within the nucleus, and then mitochondrial DNA that's contained completely within the mitochondria, just like it sounds. So there's going to be nuclear DNA and mitochondrial DNA. Nuclear DNA you inherit from both of your parents, but mitochondrial DNA you only inherit from your mother. Eukaryotic organisms do, in fact, have specialized structures called organelles. So you can think of organelles as kind of the compartments or components, organs of the cell. And there will be 3 organelles that will be important for our purposes here in this class. It'll be the nucleus, because it contains all of the nuclear DNA, which makes up our chromosomes and our genetic code. The mitochondria, because it contains the mitochondrial DNA. It's also the powerhouse of the cell. I know you guys are probably sick of hearing that, but for our purposes, we're actually more interested in the fact that the mitochondria carries the mitochondrial DNA, which allows us to trace the matriline or trace the mother's line. And we see evidence for eukaryotic organisms going back as far as about 1.2 billion years ago. So planet itself, 4.6 billion. First prokaryotic life, 3.5 billion. First eukaryotic life, about 1.2 billion years ago. All right, so this nice pretty picture here, this is a picture of a eukaryotic cell. So that nice purple mass in the middle there, that purple mass is the nucleus, which is, you kind of think of it as the brain of the cell, basically. So the brain of the cell is going to, it's going to contain all of our genetic code. It's going to contain all 46 of our chromosomes. And our chromosomes are basically DNA that's coiled around proteins called histones. So the nuclear DNA is essentially it's our blueprint for life. It contains all of the instructions for our physical structures, and it essentially is, that's why we care about the nucleus here in this class, because we are going to learn about Punnett squares and how to calculate genotypic frequencies. So that will become important later on in this chapter. I'm going to go ahead and take a little break right now because I saw that student services just popped in. So hold your thoughts on eukaryotic cell structure for a moment. And I'm going to hand it over to Gregorio. Looks like he's still connecting to audio. Hi, Gregorio. Mr. Martinez.
Hello.
Are you ready to present?
Yeah, we're ready to present. We just had technical problems, that's it, but we're here.
All right, whenever you're ready.
Mhm.
You turn on.
Yeah, turn that on. Yeah.
No, the camera.
No, the camera is on.
Hello.
Go ahead. Okay. Okay, sorry. Okay. And there we go. Okay.
Hi everyone. My name is Grigor. I'm a student ambassador at the Office of Student Life.
And I'm Gregorio. I'm also student ambassador at the Office of Student Life. And so we're here to present to you guys some ASU events that are coming up and also some benefits as well. So first up, we have our upcoming events. So we have the ASO fee, student rep fee. You might have seen this posted on our boards outside that we usually post in the campus. So if you pay a $10 ASO fee, you have access to free food at our ASU events. You get a welcome gift, get to run for office, meet with an ASU counselor here. We also have a game room, so you get to access that as well. Print 30 pages per month in the computer lab. So this is really good benefits for the $10 ASL fee, as you know, usually if you go to the library or anything like that, they charge you per the page and stuff. So instead you get 30 pages per month instead. And so yeah, these are really good benefits. So yeah. Wanna add on?
Yeah, so that's it. And you also need to pay $2 student rep fee. So it's a mandatory. So just pay it with the $10 ASO fee.
And then we also have the ASU welcome gift. So if you've paid the $10 ASL fee, as I just mentioned, you get a gift. So you get a free water bottle, free highlighter, free pen, and a free lanyard as well. And so to get this gift, you just have to come to the Student Union building, which is the yellow building on top of the cafeteria. It'll be in the second floor. So you'll just see our office right there.
On the Spring 24 campus cleanups, the location will be Student Union Plaza from 10 a.m. to 11:30 a.m. The dates are down here, so it will be on Mondays. The first one is February 26th, which is today. It happened already. And they give free food if you pay your ASO fee, the membership that we told you previous slide, like the $10 one. And then the next one will be on March 25th on Monday. And then after that, it will be April 26th. And the last one, May 13th. Yeah, if you want to see the campus clean, please come join us.
And then we also have our mural painting day. So this is tomorrow. So we'll have in the Student Union area. So downstairs where the bookstore is at and all that, we'll be having a painting day. So you get to paint a mural and it'll be from 10 a.m. to 12 p.m. Free food for paid ASL members as always. And so yeah, just come and join us to paint. And then I think they're gonna have a video, and so they'll probably show you how to paint something specific too as well. So. And then we also have our club day. So our club day is March 5th. So if you're interested in seeing clubs or meeting clubs or trying to join a club, you can come to our club days. It's from 10 a.m. to 1 p.m. There's free food as always, but the main point of this is just to get people to join clubs and come meet them, since there's new clubs that are starting out as well. So really good to just, find a spot and which club to join.
And it's also good to be part of club for your transcripts when you're planning to transfer to four-year university. And it's because it shows that you have leadership skills.
And then we also have Loteria game night. So this is it's an event where if anyone has played Loteria, or it's just like a card game where you have these boards and stuff and they call out the card name and then whoever wins, gets prizes and stuff. So we'll have that on March 28th from 3 p.m. to 6 p.m. There'll be free food as well as always. And I think they're gonna give prizes for people who win. So yeah.
Yeah, so just come and join. and ASU and DRC NORCO CSU trip. So we'll visit 3 colleges. The dates will be on April 4th, 2024 to April 6th, 2024. So it will be like 2 days trip. The application deadline will be March 1st by 3 p.m. The required mandatory meeting March 14, 10 a.m. and California Science Center. So the colleges that we'll visit will be Cal Poly San Luis Obispo, San Jose State University, San Francisco State University. So the deadline will be March 1st. So if you wanna join, please do your application.
And all travel fees are paid by ASU, so no student has to pay out of their pocket for this. So if you want to go meet these schools, take advantage of this as well. And then we also have a Mario Kart tournament. So Mario Kart tournament is on March 14th. And so this will be happening from 3:30 p.m. to 6:30 p.m. And so we'll be just having in the game room, which is on top of this cafeteria, we'll be having a tournament here for Mario Kart. So people can come in and you can win a voucher as well. And it'll be free food. So yeah, just in case you want to come and have some fun with other people as well. And then this is just the list of like the finance meetings and executive council meetings and then club council meetings and stuff for ASU. But there's also the club days posted. So just in case you can't make it for the March 5th one, we also have the one for April 6th, 16th, and then May 7th. So. And then we also have on March 8th, the first aid and CPR training. So we'll be having the CPR training on March 8th. And so it's in the Skywalk Conference Center. It's on a Friday. However, though, it is really helpful since if you attend this CPR training, you can also get CPR certified. So that means you'll be able to help people and you wouldn't be, how do I say it? So if you don't have the certification, you cannot perform CPR on people, but if you have it, you can perform it. So, right.
Yeah, exactly. So the location will be Skybox Conference Center from 10 a.m. to 1 p.m. Yeah, so the last day to sign up is March 1st, 2024 at 12 p.m. If you are interested in joining student union government, it's a beneficial of that. So you'll be involved in campus, you will become a leader. So and also you will get monthly stipends, and it is also good for your transcript because basically you are if you are part of student government, you are overlooking all the clubs and you are getting chance to travel and visit some states with the group, and you will have opportunity to meet with the higher position people, you know, like some government people, and talk to them, bring the word from college students to them, you know, and consult about that. So yeah, and it has a lot of benefits to be part of student government.
And then don't forget to follow our socials. We have our Instagram. So we have LAVC Student Life and LAVC ASU. We usually post anything that's events coming up or any information for you guys. And as well, we post any resources that are that are really helpful for any student out there. So we post that on our social media. And then we also have our Canvas shell. So we as well post everything that is coming up on there, just in case you want to stay updated. So you can scan those or you can just follow us on social media there.
Exactly. Thank you for listening.
And that will be all for us as our presentation is done here.
Thank you, Professor.
Thank you, Professor.
Oh, we can't hear you.
Sorry, I was on mute. Thank you so much, Gregorio. Thank you very much for the presentation. I have an in-person class on Monday, Wednesdays at 9:40 if you guys want to come to that one as well.
We'll message you. We'll message you.
Okay. All right. Sounds good. All right, you guys have a good day. All righty. Okay, so now we're going to shift gears back to cells. I'm also an advisor for the TAE club. If anybody's interested in becoming part of that club, hang around after and talk to me, and I can tell you more about it. All right, so let's go back here to the cell. Okay. So we were talking about the nucleus. So the nucleus is basically the brain of the cell. It contains all of the nuclear DNA, which is essentially our chromosomes that carry our genetic code. The mitochondria is this red oblong kind of oval shaped organelle. It is the powerhouse of the cell, as we all know, but it is also the holder of the mitochondrial DNA, which is um, essentially why we care about it here in this class, okay? We will learn why mitochondrial DNA is important later on in this lecture. And then the other organ I want to point out to you guys is the little blue dots on the endoplasmic reticulum. Those are the ribosomes. So the ribosomes are basically going to be the readers, and that's going to be important when we learn about protein synthesis. So the ribosomes are going to read our genetic code. All righty. Okay, so let's talk about classes of cells. We talked about types of cells, so now let's talk about classes of cells. So classes of cells, so now we're looking at just exclusively eukaryotic organisms. So we're looking within a eukaryotic organism, there are two types of cells or classes of cells, sorry. There are somatic cells and there are gametes. So somatic cells, another name for somatic cells is body cell, essentially. So a somatic cell could be a tissue cell, it could be a liver cell, it could be a blood cell, it could be a hair cell, it could be a saliva cell. You guys get the picture. Basically, any cell in your body that is not a sperm cell and is not an egg cell is going to be a somatic body cell. So it makes up the cells within our body structures, our tissues, our organs, our nerves, our bones, our skin, etc. And somatic cells are always going to be diploid. So diploid is abbreviated as 2N. Show you what it looks like. I can show you what it looks like in the chat. So when you're abbreviating diploid, it's going to look like this. Diploid is 2N. And then the N number in humans is always 23. So when you do 2 multiplied by 23, it gets you to 46. Okay, so all somatic cells within the human body are going to be diploid, meaning that they contain a complete set of your genome, all 46 chromosomes. Gametes are going to be haploid. So the way I remember it, diploid, double, haploid, half. Diploid, double, haploid, half. So gametes are going to be haploid. So a gamete is another name for a sex cell. So the female gamete is the ova or the egg cell. The male gamete is the sperm cell. So each gamete is going to contain 23 chromosomes. So all of us, essentially, genetically speaking, we inherited 23 chromosomes from dad and 23 chromosomes from mom. Of course, it's going to get more complex than that. Some genes are dominant over other genes. Some characteristics are influenced by multiple genes on multiple chromosomes. But essentially, genetically speaking, we inherited half of our genes from our mom and half of our genes from our dad. And we will learn once we get to meiosis, probably on Wednesday, when we get to meiosis, we'll learn about genetic reshuffling. So every single human has the capacity to create over 1 million different unique gametes. So even if you have the same mother and same father, you don't necessarily look identical to your biological siblings. Because even though you have the same mother and same father, each time that sperm and egg cell is produced, it goes through the process of crossing over and genetic reshuffling. All right, so a zygote is essentially a fertilized egg. So once the sperm cell fertilizes the egg cell, you get a zygote. It's still not, the zygote still has to travel through the fallopian tubes and then eventually attach to the uterine wall. So not all zygotes become developing pregnancies. It's not always a sure thing. But a zygote is essentially once that sperm cell fertilizes the egg cell or the ova. So 23 chromosomes from mom's ovum and 23 chromosomes from dad's sperm makes a zygote. Questions? Nothing? All right, let's shift gears just a little bit. Now we're going to focus in on DNA. So there will be 2 important processes that we learn today with DNA. We're going to learn about DNA replication and also protein synthesis. But first, let's talk just a little bit about the structure. So we talked about the structure of the cell. Let's talk a little bit about the structure of DNA. So like I mentioned, DNA is essentially our chemical template or our blueprint for life. It's the chemical template for every aspect of an organism. It codes for all of our physical attributes, what we look like, the structure of our bodies, of our organs, what color hair we have, what color eyes we have. It's a chemical template for every aspect of who we are as an organism. Now, of course, like we mentioned on the first day of class, Humans are complex. We're more than just our biological makeup. We are also influenced by our environment, by our culture, um, by our rich heritage, by all those things. But at least part of who we are is influenced by our chemical template. So DNA in a eukaryotic organism is in a double helix ladder-like structure. So when you look at this image here at the bottom, it's a microscopic image of DNA. It looks like a ladder that's been twisted, right? It's a twisted up ladder. So the sides of the ladder, those are made up of alternating sugar and phosphate molecules. And then the rungs of the ladder, essentially where you'd be stepping if you were climbing up this ladder, the rungs are made up of alternating nitrogen base pairs. We call them complementary nitrogen base pairs. So in DNA, adenine will always pair with thymine and vice versa. And cytosine will always pair with guanine and vice versa. So it's always going to be A to T and always C to G. All right, so the components of a nucleotide. So a nucleotide on the sides of the ladder. So the sides of the ladder is alternating sugar and phosphate molecules. The sugar molecule, the type of sugar will determine whether it's DNA or RNA. DNA gets its name from the sugar called deoxyribose. So that's where we get the D in DNA. And RNA has a sugar called just ribose. So that's where we get the R in RNA. So these nucleotides are essentially what makes up the double helix-like structure. So we've got our sugar molecule, we've got our phosphate molecule on the sides of the ladder, and then, like I mentioned, the rungs of the ladder, what you would step on, those are made up of the alternating complementary nitrogen base pairs. So it's adenine pairing with thymine, guanine pairing with cytosine, and vice versa. All right, so there are two different types of DNA. There is nuclear DNA, which is contained completely within the nucleus of the cell. And this is what actually makes up our actual genetic component. That's what actually codes for our physical attributes, our physical features, and things like that. So the nuclear DNA will coil around these proteins called histones to make visible chromosomes. And in every somatic cell, so every skin cell, every saliva cell, every tissue cell, every bone cell, we have a complete set of our genome. So that's why when you're watching forensic files and you see they get just a little bit of saliva or some skin cells, and they're able to determine who that individual is. Of course, if they have the funding and if the DNA didn't degrade, but if they were able to successfully extract even just a little bit of saliva, without contaminating the DNA, that scientists could potentially determine the genetic makeup of that organism, of that person. So nuclear DNA is what we call homoplasmic. So the root of that word, homo means same, plasmic means blood, DNA. So it's referring to the nuclear DNA, which is identical in the nucleus of each cell type. Now, of course, cell types will have instructions. Some cells become organs, some become tissues, some become blood, some become bones. You guys get the picture. But essentially, every single somatic cell will have a complete copy of the genome within the nucleus of that cell. That's what nuclear DNA looks like underneath a microscope in the chromosome form, the visible chromosome form. All right, and then the other type of DNA that I mentioned earlier is the mitochondrial DNA. So you might sometimes see mitochondrial DNA abbreviated as lowercase mt and then uppercase DNA. So if you see that, just know that that's just shorthand for mitochondrial DNA. Mitochondrial DNA is contained mainly within the mitochondria, just like it sounds. There's a little bit in the cytoplasm, but it's mainly in the mitochondria. And like I mentioned before, the reason we care about mitochondrial DNA is because in evolutionary terms, it allows us to trace what we call the matriline. So your mother's 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 mother. You get the point, right? All the way back to, they've traced it back, some studies have traced it back to hundreds of thousands of years, looking for that mitochondrial Eve, essentially. But we will talk more about this when we get to the human evolution sections. We'll look at we'll look at nuclear DNA studies versus mitochondrial DNA studies. We can also trace markers on the Y chromosome if we're more interested in the patriline. But pretty cool stuff. But that's essentially why we're kind of doing this chapter on introducing DNA in an anthropology class, because we will eventually get to chapters where it's going to be useful to at least have a base level understanding of nuclear DNA and mitochondrial DNA. All right, so like I mentioned before, DNA is going to be responsible for two vital processes. So the first one we're going to talk about is going to be just straight up DNA replication, which is going to happen as kind of the prep phase of cell division, of mitosis and meiosis. And then protein synthesis we'll probably get to today as well. So protein synthesis is a little bit more complicated, has a few more steps, but it's important because it's essentially the way that we read, the way that our genes are read, essentially. All right, let's talk first about DNA replication. So we know that DNA replication is going to occur during interphase or the prep phase of both mitosis and meiosis. So we'll learn more about mitosis and meiosis probably Wednesday, but just know for now that mitosis is necessary for growth and tissue repair. So if we have a scrape on our arm, then our skin cells need to regenerate themselves. So and each skin cell, every new skin cell is also going to need a full set of our genome. So that's why DNA replication has to occur during the prep phase of cell division, because both the parent cell and the daughter cell are still going to need that set of DNA. So that's why we need DNA replication. So this particular process, DNA replication, will occur entirely in the nucleus of the cell. It's going to involve the parent strand essentially unzipping. There is an enzyme called DNA helicase that will inspire the parent strand to unzip and expose all of the nitrogen base pairs. So in this image here, the blue ribbon, that's representing the parent or the old strand. We call it the old strand or the parent strand. And then that gold one coming in, that's representing the new or the daughter strand. So you see this unzipping process, you can kind of see here that it's exposed all of those T's, G's, C's, and A's. So it's exposed those nitrogen base pairs. So then the daughter strand comes in and pairs up following those rules of adenine pairing with thymine and vice versa, and cytosine pairing with guanine and vice versa. So that's why it's important that we memorize that, because there will be, some exercises where it has you kind of recreate a DNA strand or kind of, basically simulate what's happening during DNA replication. So I'm going to show you guys a little video clip. So for this chapter, I really like to use a series called Amoeba Sisters. And you guys can tell me if it's too cartoony. But I like this particular series because it does, kind of give you a visualization, and it really breaks things down. Because I know it may be a while, it may have been a while since some of us have taken biology. So I kind of like to show you guys a little visualization of what this might look like. So you guys tell me if this is too simplistic. I do have other videos I can show you. But let's just pull it up here. Sorry, I should have gotten this pulled up ahead of time. It's running a little bit behind today. All right, so Amoeba Sisters. DNA replication. There we go. All right, there it is. Okay, so go ahead and just sit back and enjoy, and we will talk about it after. With these, biological concepts, don't feel that you need to memorize or write down every little detail. What I want you to know is the big picture. So think about, what are the major steps and what's the end result, basically.
That introduces our DNA. We talk about it so much. It's the ultimate director for cells, and it codes for your traits. With a molecule that has a function like that, it makes sense that when you make another cell, like in cell division, you would also need to get more DNA into the new daughter cell. And that introduces our topic of DNA replication, which means making more DNA. First, let's talk about where and when. First, where? Well, if it's in a eukaryotic cell, it occurs in the nucleus. However, remember, not all cells have a nucleus, such as prokaryotic cells. They don't have a nucleus. Still, both prokaryotic and eukaryotic cells do DNA replication, but there's some differences between the two that this clip doesn't go into. Next, when? When does this happen? Well, a cell is going to need to do this before it divides so that the new daughter cell can also get a copy of DNA. To get specific, in a eukaryotic cell, That's going to be before mitosis or meiosis in a time known as interphase. I think DNA replication would actually make a great video game. Still waiting for that to be invented. I'm going to introduce the key players in DNA replication so that you can get some background information. And remember, these are just some major key players. There's a lot to this process. Many of the key players are enzymes. In biology, when you see something end in ASE, you might want to check, as it's very possible that it's an enzyme. Enzymes have the ability to speed up reactions and build up or break down the items that they act upon. So here we go with the key players. Helicase, the unzipping enzyme. If you recall that DNA has two strands, you could think of helicase unzipping the two strands of DNA. Helicase doesn't have a hard time doing that. When unzipping, it breaks through the hydrogen bonds that hold the DNA bases together. DNA polymerase, the builder. This enzyme replicates DNA molecules to actually build a new strand of DNA. Primase, the initializer. With as great as DNA polymerase is, DNA polymerase can't figure out where to get started without something called a primer. Primase makes the primer so that DNA polymerase can figure out where to go to start to work. You know what's kind of interesting about the primer it makes? The primer is actually made of RNA. Ligase, the gluer. It helps glue DNA fragments together. More about why you would need that later on. Now, don't feel overwhelmed. We'll go through the basics of this sequence in order. But remember, like all of our videos, we tend to give the big picture. There are always more details and exceptions to every biological process that we can't include in such a short video. DNA replication starts at a certain part called the origin. Usually this part is identified by certain DNA sequences. At the origin, helicase, the unzipping enzyme, comes in and unwinds the DNA. Here's the thing though, you don't want these strands to come back together. So SSB proteins, which stands for single-stranded binding proteins, bind to the DNA strands to keep them separated. And topoisomerase, I always have to slow down when I say that enzyme's name, keeps the DNA from supercoiling. Supercoiling might sound super, and it can be when you're trying to compact the DNA, but it's something that needs to be controlled during DNA replication. Supercoiling can involve an overwinding of the DNA, and you need the DNA strands to be separated for the next steps. Primase comes in and makes RNA primers on both strands. This is really important because otherwise DNA polymerase won't know where to start. In comes DNA polymerase. Okay, before we go on, remember how we said DNA has two strands? They're not identical, they complement each other. In our video that covers DNA structure, we talk about how the bases pair together with hydrogen bonds. The base adenine goes with the base thymine, and the base guanine goes with the base cytosine. These strands are also anti-parallel, so they don't go in the same direction. What do we mean by direction? Well, with DNA, we don't say north or south. We say DNA either goes 5' to 3' or 3' to 5'. What in the world does that mean? Well, the sugar of DNA is part of the backbone of DNA. It has carbons. The carbons on the sugar are numbered right after the oxygen in a clockwise direction. 1', 2', 3', 4', and 5'. The 5' carbon is actually outside of this ring structure. Now, you do the same thing for the other side, but keep in mind DNA strands are anti-parallel to each other. So let's count these, again clockwise after the oxygen. 1', 2', 3', 4', 5'. And the 5' is out of this ring. This strand on the left runs 5' to 3'. And the strand here on the right runs 3' to 5'. We'll explain why all that matters in a moment. So let's take that knowledge there and look at DNA replication here. In this image, I labeled the top original strand 3' to 5'. I labeled this bottom original strand 5' to 3'. That's the original DNA that is going to be replicated. DNA is unwinding here thanks to helicase. In this example, it will keep unwinding in this direction. Primase places primers. DNA polymerase is building the new strands. Now, the thing about DNA polymerase is, when it's building a new strand, it can only build the new strand in the 5' to 3' direction, meaning it adds new bases to the 3' end on the new strand. See how it's being built in the 5' to 3' direction? This one is called the leading strand. But take a look down here. So DNA polymerase once again is building a new strand in the 5' to 3' direction. But there's a bit of a problem here. See, as DNA unwinds, because DNA polymerase can only build the new strand in the 5' to 3' direction, it has to keep racing up here next to where this unwinding is happening. You can see why then this new strand is known as the lagging strand. On this lagging strand, primers have to keep being placed in order for DNA polymerase to build. These fragments that result are known as Okazaki fragments. Primers have to get replaced with DNA bases since the primers were made of RNA. Ligase, the gluing enzyme, as I like to nickname it, has to take care of the gaps between Okazaki fragments, sealing them together. At the end of replicating, you have 2 identical double helix DNA molecules from your one original double helix DNA molecule. We call it semi-conservative because the 2 copies each contain one old original strand and one newly made one. One last thing. Surely you've had to proofread your work before to catch errors. In this process, you don't want DNA polymerase to make errors. If it matches the wrong DNA bases, then you could have an incorrectly coded gene, which could ultimately end up in an incorrect protein, or no protein. DNA polymerase is awesome. It has proofreading ability, meaning it rarely makes a mistake, which is a good thing. So remember how we said there is far more detail to this process to explore? The detailed understanding of DNA replication has led to some life-saving medical treatments that can stop DNA replication in harmful cells, including pathogenic bacteria or human cancer cells. We encourage you to explore beyond the basics. Check out the further reading suggestions in the video details to explore more. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
All right. So I know that was a lot of details. So please don't stress. I'm not expecting that you remember every detail of that. What I really want you to know is the big steps and the end result, and essentially why we care, why it's important for our purposes as biological anthropologists. So, I do want you to understand what pairs with what. I want you to understand when and where it happens. And definitely knowing that the purpose of it is so that we can have cell division and that the resulting cells can still have a complete copy of our genome. Because without that, then every time we go through mitosis or meiosis, our chromosome number would get chopped in half, which that's no good because then we're losing all of our DNA. So we need DNA replication to allow for cell division to happen. And it's a semi-conservative process. That's important. That part's important too, because your end result is they're identical strands, but they're half the parent strand and half the daughter strand, or half the old, half the new. So that's why we call it a semi-conservative process. All right, questions about DNA replication? Feeling okay? All right. Okay, so now we're going to shift gears a little bit. We're still going to be talking about DNA, but we're now going to be talking about a process called protein synthesis, which will be somewhat similar to DNA replication, but there's some additional steps that go in there. So just as a little bit of a review, if we're talking about DNA, adenine will always pair with thymine, guanine will always pair with cytosine, thymine will always pair with adenine, and cytosine will always pair with guanine. So it's always going to be A to T and C to G. Now, the only difference when we're talking RNA, the only difference is adenine is now going to pair with a new nitrogen base called uracil. And that'll be important once we get the messenger RNA and the transfer RNA involved. Okay, this will make more sense here in a moment. But in DNA, adenine always pairs with thymine, guanine always pairs with cytosine, thymine always pairs with adenine, and cytosine always pairs with guanine. The only difference in RNA is adenine will now pair with a new nitrogen base called uracil. Okay, so protein synthesis. So we care about protein synthesis because this is essentially how the ribosomes are going to read our genetic code. So proteins are essentially chemicals that make up our tissues. Proteins can also regulate functions, repair, and the growth of tissues. Proteins are made up of something we call amino acids, or a chain of amino acids. There are 20 different amino acids, and these amino acids are organic molecules that combine in a specific sequence by the ribosomes to form a protein. So that'll be one of our steps in our process, basically. So essential amino acids, those are the ones that you get exclusively from, or those are the proteins that form an organism's physical attributes. And those are also ones that you get from your diet as well. Okay. All right, so just like there's types of cells and classes of cells and types of DNA, there's also types of proteins. So there are structural and regulatory proteins. So the structural proteins are responsible for our physical characteristics. So some examples would be keratin and collagen. These are proteins, structural proteins that are found in both our skin and our nails. And then regulatory proteins, just like they sound, they regulate things. They're responsible for bodily functions. So things like enzymes, hormones, antibodies, those are all considered regulatory proteins. So protein synthesis, and we're going to break this down a little bit more, but protein synthesis will involve two primary steps. The first step is called transcription, which is going to begin in the nucleus. It starts out just like protein, it starts out just like DNA replication with the parent strand unzipping and making a template for the RNA. So that messenger RNA strand will then go out into the cytoplasm where the ribosomes are located, and those ribosomes are going to read our genetic code codon by codon. So a codon is going to be a set of 3, like AUG, for example. AUG is the start codon. Okay, let's talk about each in a little bit more detail. So the first step, called transcription, is going to occur entirely in the nucleus of the cell where the DNA resides and will eventually result in the formation of a messenger RNA strand. Okay, so that when you see that abbreviated like that, lowercase m and then RNA, that's referring to messenger RNA. Okay, so the messenger RNA is necessary because DNA is too large, because it's double-stranded, it's too large to leave the nuclear pore. And it needs to leave the nucleus because it needs to go out to the cytoplasm where the ribosomes are located. So the reason we need RNA is because we need a single-stranded molecule that's small enough to leave the nuclear membrane and go out into the cytoplasm where the ribosomes are located. Okay, so the end result of transcription is the formation of a messenger RNA strand. Okay, so during step 2, the second step is called translation. So that messenger RNA strand will move out into the cytoplasm where the ribosomes are located, and it will essentially translate into a specific order of amino acids. So this will make more sense when we watch a little video clip, but just think about the ribosomes are essentially the readers. The ribosomes are going to read 3 by 3. So think of each codon as a word. So each set of 3, like AUG, for example, and then, CAT, something like that. It's always going to be read in sets of 3. So each set of 3 is going to then code for a specific amino acid. Okay, so here we are in this step here. So the ribosomes are reading the messenger RNA bases 3 at a time, codon by codon. And then the transfer RNA is going to bring in the amino acid into place. So imagine the transfer RNA just bringing that amino acid into place, and then we're creating this polypeptide chain. So breaking that word up, polypeptide, poly means many, peptide means proteins. So we've got this chain, this polypeptide chain of amino acids that is coding for a specific protein. Okay, so one more time, we've got transcription that's occurring in the nucleus of the cell. It starts out just like DNA replication, and it involves the helicase unzipping the DNA strand, exposing those nitrogen-base pairs. But we've got this single-stranded messenger RNA that's small enough to leave the nucleus and travel out into the cytoplasm. So the mRNA leaves the nucleus, travels out into the cytoplasm where the ribosomes are located. And then step 2, known as translation, is going to occur entirely out in the cytoplasm where the ribosomes are located. So the messenger RNA carries its message of the sequence of DNA bases And the ribosomes, the readers, are going to go through and read the code codon by codon, or set of 3 by set of 3. So then the transfer RNA, tRNA, is going to bring in the appropriate amino acid and lock it into place. So the end result, we've got this chain of amino acids, also called a polypeptide chain or polypeptide sequence. Okay, I know this is a lot of detail, so let's try to visualize it again. Were you guys okay with the Amoeba Sisters, or is it too cartoony for you?
No, it's fine.
Okay, all right, let's watch the Amoeba Sisters again. And again, just kind of sit back. Don't feel like you need to memorize every detail. But what I want you to do is look at the major points. Look at the, what are the big steps? Look for the big steps. What's the end result? And essentially, why do we care? Why is it important for us to understand protein synthesis in a biological anthropology class? So think about, what's the big picture? All right, let me pull it up here. Oh, it's right here on this side.
After learning about DNA, have you ever wondered how can the DNA actually result in a trait? Let's take an example, like eye color. Yes, your DNA has the genetic information that codes for the color of your eyes. Your eye color is based on a pigment that is inside the eyes, but in order to have that pigment, you have genes, which are portions of DNA that can code for proteins which help make that pigment. So what we're going to talk about is how your DNA can lead to the making of a protein. This process is called protein synthesis. Synthesis essentially means to make something. So protein synthesis means to make protein. And you may wonder, what's the big deal about proteins? Well, you may not realize this, but proteins are kind of a big deal. They do all kinds of things. Proteins are involved in transport, in structure, in acting as enzymes that make all kinds of materials, in protecting the body, and so much more. You've got to make proteins. It's essential for you to live. And what is so cool is that you are making proteins right now as you sit and watch this video. It's happening in your cells. They're making proteins. So back to your DNA and its role in all of this. All of your cells have DNA. Well, a few exceptions. And that DNA is in the nucleus. Some DNA is non-coding DNA. Some DNA makes up genes that are not activated. More about that in our gene regulation video, but we're going to talk about genes that are coding for active proteins. So how are we going to get the information from these genes out of the nucleus so that the cell can start producing the proteins that it needs to make? Well, let us introduce you to the amazing work of RNA. We have a video comparing and contrasting RNA and DNA. We'll be brief here in saying that RNA is a nucleic acid, like DNA, but it has a few differences. Its role in protein synthesis also is huge. Before we get into the process, please note our typical disclaimer. We tend to simplify topics, but as always, we hope that you have the desire to explore this complex, amazing process later on, to learn all about the extra information that we don't have the ability to include in this short video. In protein synthesis, we can look at two major steps. One is transcription, and the other is translation. Transcription has a C in it, and translation has an L in it. I remember that C comes before L in the alphabet, which helps me remember that transcription comes first. I like a lot of alphabet mnemonics. Now, transcription is when we're going to transcribe the DNA into a message. In your cells, the DNA is in the nucleus, so therefore we're doing transcription in the nucleus. In the step of transcription, an enzyme called RNA polymerase will connect complementary RNA bases to the DNA. These RNA bases are bonded together to form a single-stranded mRNA. The M in mRNA stands for messenger. Messenger RNA consists of a message made of RNA that has been based on the DNA. We do want to mention that this mRNA is not usually ready to go right away. There's usually a significant amount of mRNA editing that occurs. We highly encourage you to do some reading about that because it's not only fascinating, it's critical for the process to work correctly. So what's something great about being mRNA? Well, in eukaryotes, you get out of the nucleus. The mRNA can go out of the nucleus into the cytoplasm where it's going to attach to a ribosome. Ribosomes make protein. The ribosome is made of rRNA, and that's an easy one to remember because the R stands for ribosomal RNA. The ribosome is going to build our protein in the next step called translation. You know, you can find a lot of great clips and animations on translation that are just fantastic. We're just going to break down some basics of what's happening. In the cytoplasm, if you look at this, you have all these tRNA molecules available. tRNA stands for transfer RNA. They carry an amino acid on them. An amino acid is the monomer for a protein. It's a building block for protein. Since we're making proteins, we're going to need those amino acids to build it. If you have a bunch of amino acids together, you can build a protein. So it's the tRNA that is going to bring those amino acids together to make that. But wait, how does the tRNA know which amino acids to bring? That's why the mRNA, the message, is so important because it's going to direct which tRNAs come in and therefore which amino acids are transferred. All of these tRNAs are looking for complementary bases. When they find the complementary bases on the mRNA, they transfer their amino acid. When tRNA is bringing in the amino acids, it reads the bases, represented by these letters here on the mRNA, in threes. So it doesn't read one letter at a time, it reads in triplets. That's called a codon. So for example, in this mRNA, the tRNA would read the codon AUG. One of these tRNAs contains a complementary anticodon, which in this case is UAC. All tRNAs that have the anticodon UAC will be carrying an amino acid called methionine. A tRNA with the UAC anticodon comes in to pair with the complementary AUG codon on the mRNA. It transfers the amino acid it carries, methionine. The tRNA will eventually leave, but it will leave behind its amino acid. That's the first amino acid before looking at the next codon. Before we do the next codon to carry this on, if you're wondering, how did you know that the tRNA that went with the AUG codon would be carrying an amino acid called methionine? Well, for that, you will find a codon chart helpful. You can learn to use a codon chart to determine which amino acid each mRNA codon will code for. Isn't it so fascinating that scientists have been able to determine which amino acid corresponds with these codons? I used to have a codon chart poster and just marvel at that. You could see on a codon chart that the AUG codon on the mRNA codes for methionine. AUG is also considered a start codon, as methionine is typically going to be your first amino acid in proteins. There are many types of amino acids in the codon chart, but there are even more possible codon combinations. That means there can be more than one codon that code for the same amino acid. For example, according to the mRNA codon chart, all of these mRNA codons here code for the same amino acid, leucine. That means their complementary tRNAs all carry the same amino acid, leucine. Okay, so going back to the mRNA, let's try the next codon on this mRNA, CCA. On the codon chart, you can see that codes for the amino acid proline. The complementary tRNA has the anticodon GGU. And look, there's the proline that we knew it would be carrying. The tRNA will transfer that amino acid and eventually leave where it can pick up another amino acid. These amino acids are held together by a peptide bond, and it will keep on growing. Typically, at the very end of the mRNA, there is a stop codon. Stop codons do not code for an amino acid, but when the ribosome reaches it, it indicates that the protein building is finished. So the result of translation is that you built a chain of amino acids that were brought in certain sequences based on the coding of the mRNA. But remember, that mRNA was complementary to the DNA, so the DNA ultimately was the director of the entire protein building. Of course, it couldn't have done it without some serious help from mRNA, rRNA, and tRNA. Protein folding and modification may occur, and the protein may need to be transported. This can all vary based on the protein's structure and function. Another fascinating topic for another Amoeba Sisters video. Well, that's it for the Amoeba Sisters, and we remind you to stay curious.
All right, I know that's a lot of details, but what I want you guys to do, I want you to go into your breakout rooms, and I want you to describe the process. So start out with DNA replication and try to describe the process in, oh, I don't know, 3 to 4 sentences or bullet points, however you want to do it. And then do the same thing for protein synthesis. So it's totally fine to use your notes, totally fine to go back and look at the textbook or whatever you need to, or watch the video again. But I want you to try to summarize both processes in about, I'd say, 3 to 5 bullet points or 3 to 5 sentences, however you'd like to do it. So imagine exactly how you would explain the process to somebody not taking the class. All right, questions.